Thank you so much for the introduction. So today I'm going to talk to you guys a little bit about the work that my laboratory has done over the course of the past 8 or so years. I'd like to preface it by saying that I have been a virome and microbiome guy for many, many years. And my laboratory has just sort of started off doing some more basic phage and phage therapy sort of work. So then most of what I'm going to show you guys today is all sort of describing and understanding the viruses that inhabit the human body and what role they may be playing in the human microbiome. So I'd like to thank everyone for coming. June is not an easy month as we had a hard time finding a speaker, thus I ended up being the speaker, but I really appreciate everyone coming. So I want to take you guys through sort of some of the history of the things that we found in the human virome, and I think probably closer towards the end it'll become really obvious how my laboratory sort of got interested in looking at bacteriophages as well as interested in issues related to phage therapy. So we have really considered ourselves to be a microbiome laboratory. That's what I have been telling people for the past 8 or so years. We're just a microbiome focuses on viruses. And that's really been a hard sell for much of the time that I've been doing this, in that, I tell people I work on the human microbiome and they immediately think, oh, those bacteria that live in and on your body. And then I sort of have to correct to say, hey, the human microbiome is really a lot more than just those bacteria that inhabit the human body. There's a lot of other things there as well. So bacteria, for obvious reasons, given some of the findings, are the first thing that popped to people's mind. But there's really a lot of viruses that inhabit the human body. Whether you're healthy or whether you're diseased, you're carrying around a lot of viruses. And of course, there are a lot of other microorganisms in the human body as well. And my laboratory was really one of the first to get started in human virome research. And when we started, some of the questions we would ask about the human virome were basically the exact same questions that were being asked about the human microbiome. So for example, 15 years ago when human microbiome studies really started, the biggest question was always sort of, where does the microbiome live? Those are questions you don't see asked anymore. And the main reason is that the answer was pretty much probably everywhere. And we have gone on to show over the course of years that all of these same parts of the human body that are inhabited by these bacterial communities are also inhabited by viruses. In particular for MDs like myself, who have been taught certain things about microorganisms in the human body. So for example, when I was taught about urinary tract infections, I was taught that your urine is completely sterile unless you have an infection. If you don't have pneumonia, your lungs are completely sterile. And a lot of these studies of the human microbiome as well as the human virome have really shifted the way we think about it. Because I can tell you the urine is absolutely not sterile, whether you've got an infection or not, your lungs are not sterile, et cetera, et cetera. So I think the bottom line is that we are full of microorganisms. The most recent estimates of the human body is that we've got about 30 trillion of our own cells. Compare that to the fact that most human bodies are going to have about 38 trillion bacteria. And one of the main reasons that we're really interested in viruses is that we estimate that there's 10 times as many viruses in the human body as there are bacterial cells. So we estimate there's about 380 trillion viruses in the human body. And one of our big goals is basically to discover what they are and what they're doing. So most of the focus of a lot of the microbiome studies you guys have probably seen over the years are, what bacteria are in the body, where are they located in the body, how do they respond to disturbances such as antibiotics, and ultimately how might they affect human health and disease. Those are all the same questions that we're asking. We're just taking a virus-centric viewpoint. And one of the points that we sort of found out really early on in our study of the human virome is that most of these viruses that we can identify in the human body are actually bacteriophages. So that really has shaped sort of the way we think about it. So we've got sort of two major hypotheses that permeate throughout all of our work when we're looking at the human virome. First is that if you think about all of the studies that sort of show that bacterial communities play a role in health and disease through various mechanisms, if the viruses in these communities are changing the bacterial communities, then viruses must also play a role in human health and disease. So we're trying to discover what that role is. The other is that viruses are shared. And when most people think about viruses, they're thinking about things like the flu and the common cold and so forth, things that are really contagious. While we don't really think about viruses in your virome as being contagious, we know, and I'll show you data, that indicates that they're really easily shared, which means for me, if my viruses are shaping my bacterial communities and potentially determining health and disease in me, if I share my viruses with you, then my viruses are potentially determining health and disease in you as well. So we're really trying to figure out what is the role of viruses, particularly when they're shared. So when we started out in human virome research, we had a lot of very sort of basic questions, you know. One of the first questions I asked is, are viruses transient members of your microbiome? Or, for example, are the same viruses you have today the exact same ones you're going to have a year from now or even 10 years from now? We really had no idea about that. Do these classically sterile sites like urine and the lungs actually have viromes? How do viromes differ between two different body sites? In other words, are the viruses in your mouth the same as the viruses that you might find in your gut? Are viruses shared? And we'll show you data to indicate that they are. And then lastly, and what we'll end up on, is how do we measure the effects of viruses in your bacterial community? So we'll kind of go through a bit of each of those. We started out our virome research looking in the human oral cavity. We were a laboratory that was really interested in periodontal health and periodontal disease. And when we first started out these studies, we were really sort of naive about viruses in the human body. And it was a really sort of nice time to study the human virome, because at the time, we didn't really even know that the human virome existed. So I remember some of the first questions I was asking when I entered a lab that was interested in the human oral cavity is, are there viruses in the mouth? You know, it seems like a really sort of simple, naive sort of question, but it's actually the question I was asking, right? It's obvious if you had like a herpes or a cold sore that you had a virus. It wasn't obvious if you were healthy that you might have viruses in your mouth. So the first study that we did way back when was a study that, quite frankly, in retrospect, seems like it was doomed to fail because this experiment is just me and a bunch of people I was able to round up in the lab spitting in a tube. And all we did was spit in a tube. We took it downstairs, we stained them, and then we looked at it under electron microscope. If any of you guys have ever tried to do things like this under electron microscope, you know, in general, that's not very easy to do, but that's exactly what we did. So I'm actually subject number one here, and these are viruses that are present in my mouth. And while that experiment was so poorly designed and seemed like it was doomed to fail, it was actually destined to succeed. And the reason it was destined to succeed is that there were so many viruses there that anybody could have found them. So this experiment actually worked out quite well, even though if I were training a graduate student nowadays, I would tell them absolutely don't do something as stupid as this. So we worked really hard then to figure out what are all of these viruses. And in fact, I came down here, my first trip ever to San Diego, I spent a week in Forrest Rower's lab, and he actually taught me how to look at and analyze these viruses. And the one thing I should say about virome research in general is that the one thing that we try really hard to do is to get rid of all of the human cells, get rid of all the bacteria, so that we're looking specifically at viruses. And it's sort of a very lossy process. In other words, we centrifuge them, we do all sorts of different things, and you end up losing all the big stuff, losing all the really small stuff, and we're only looking at DNA viruses. So that when we say we're looking at the virome, we're looking at a good portion of the viruses that are there, but we're definitely not looking at all of the viruses that are there because of the process that it takes to actually get these viruses out. But we were really shocked. when we saw the results of these initial experiments, the main reason we were surprised by that is that my goal was to work on human viruses. You know, I was an infectious disease doctor. I wanted to work on, you know, all these fun viruses like flu and Ebola and HIV and so forth. So I thought that we were going to be looking at human viruses. When we did these experiments, it turned out that over 99% of what we found were all bacteriophages. And in retrospect, it's really, it was a big surprise back then, but now it just makes perfect sense, right? If you've got more bacteria in your body than you have your own cells, you're probably going to have a lot more bacterial viruses in your body than you have your own cells. So it's nice to kind of go back and look at our mindset to see just sort of how naive about these things that we are. So I knew nothing about phages at the time. But the simplest thought process is that phages kill bacteria. And at this time, we knew that most of the bacteria that you have today are the same bacteria you're probably going to have in a few months from now, unless, for example, you take antibiotics or have some big disturbance. So I came up with this really simple model system for how I thought viruses probably behave in the human microbiome. And that is that a virus comes into the system, identifies a host, kills off that host, the host develops resistance, and it recovers. So that the host is always sort of semi-stable, but that because that resistance develops, the viruses always disappear. So a virus will come in, it will make progeny and disappear, and another virus and another virus. That's what I thought was probably the way that these systems work. So when I first started off my research laboratory, the first experiment that we set up was actually to demonstrate that viruses were transient members of the human microbiome. And the way that we did that was we took 8 subjects. We looked at them at 11 different time points over a course of 60 days and basically just had them all spit in tubes and froze it, and then looked at the viruses that are present in their saliva. The reason we chose saliva is that, you know, saliva is very transient. You make it, you swallow it, you spit it out, but usually the same saliva that's in your mouth right now is not going to be the same saliva that's in your mouth at the end of the day. So we thought it would be the perfect sample type to look at to show that viruses were transient. So we sequenced all of these viruses and we put together this heat map. And as you guys can see, this heat map looks like a matrix. Was never intended to look like a matrix. Whatever the opposite of a matrix is what we intended it to look like. But it looked like this because the same viruses were enriched and present in every single subject. So in fact, it's looking like viruses actually are persistent members of your oral cavity or the human microbiome, not transient members of your microbiome. And just to give you guys further details as to how sort of stable this is, we took all the sequences from all of these time points and assembled contigs, and these contigs are contributed to fairly equally by every single time point, which is telling us that the same things basically are being made regardless of what time point you look at, that these viruses are basically remarkably stable members of your microbiome, whether you're looking at day 1 or you're looking way out at 60 days. We have quantified how persistent these viruses are. So for example, here we're looking at viruses that are present greater than 7 days versus viruses that are relatively transient for just a few days. And we can see that there are significant differences. About 70% of these viruses we could find in your saliva at every single time point that we looked at. We've even gone on to look at that in the GI tract as well, and we find, again, right around 70% of the viruses in your GI tract you can find it for 7 days or more. So again, we set out to show that transience was the rule. We actually found that transience of viruses is the exception, and that persistence appears to be the rule. And in fact, when we took some of these and did good old-fashioned Sanger sequencing of these viruses at every single time point just to as a further proof that they're present. And we can not only find them present, but we can find mutations that are present as well. And while we typically think of viruses as, they have these high mutation rates, they're constantly changing all the time, the one thing that we found is these viruses weren't changing very much at all throughout the course of this study. One of the main reasons is that a lot of these viruses are lysogenic or temperate, meaning that they live in the bacteria's genome, and a lot of their sequences sort of follow the bacterial genome rules in terms of mutation rates and not typical virus polymerase mutation rates. One of the other things that we looked at is how viruses differ between different individuals. And while this is only shown in a 2D form, if you were to look at this in 3D form, it'd be obvious. Every single one of these subjects, regardless of which sort of time point you're looking at, is different from one another, so that we can tell exactly who you are just by having you spit in a tube. I mean, it's an expensive thing to do, so that we wouldn't really want to do that. But if we had to do that, we could do that just by looking, for example, at the viruses that are present in your mouth. And we can even look at, through some of these tests, we can even show that your viruses are individual specific to the point where some of these reach the level of statistical significance or pretty close to statistical significance, depending on how you do the test. One of the other things that we have demonstrated is the idea of individual specificity, but also the concept of sharing your viruses. So, for example, we went on to do what we call the campus virome study, where we actually took roommates on a campus as they were just moving in together and looked at them over the course of 6 months and just sort of asked the question, what do your viromes look like? Here we're looking at the gut, but the gut results are basically the same that we can see by looking in your mouth as well. At every single time point, we could tell subject 1 apart from subject 2. So again, the viruses really paint a nice picture, particularly when you look at them longitudinally, of exactly who you are, and we find it in pretty much every study that we do. But when we look at these viruses in a different context, we start to see these household patterns emerge. And these household patterns did not take long times to develop. Within a few days, we start to see these household patterns start to emerge. So not only can we tell who you are by looking at the viruses in your mouth or in your gut, but we can tell who you live with just by looking at the viruses in your mouth and your gut. And we believe that all this is taking place because you share your viruses. I do want to mention that most of these viruses are bacteriophages, so a lot of what's actually being shared are bacteriophages. And we can also show that this sharing is very household specific, and we can show that it's statistically significant in 7 of these 8 households, with a huge trend even in the one household that was not statistically significant. So these aren't things that happen by chance. This is real evidence of people sharing viruses in short time periods where these viruses really are taking hold within their microbiomes. We've gone on to do, again, some more traditional Sanger sequencing, where we can actually demonstrate, hey, we can do PCR and we can find each of these viruses in one housemate, and then we can do PCR and show that same virus appearing in that person's, in that housemate's virome as well. When we do this sort of analysis, right around 25% of these viruses look like they're shared between household members, and right around 10% of these have patterns that's consistent with being transmitted from one household member to the other household member over the course of the study. Not only is there sharing between unrelated roommates, but there's also sharing between genetically related individuals. We looked, for example, at just breast milk between mother and child. So we did this for a couple of reasons. One is that we're interested in how the infant virome forms. The other is we were interested in showing that there are viruses present in maternal milk. So we know there's viruses from this study present in maternal milk, but also when we do this simple PCL-OA plot, we can see huge differences. For example, we just compared them with like adult stool, adult urine, and saliva. But on this other side, we see this sort of hodgepodge of viromes which represent mother's milk as well as the infant stool. Certainly looks like there's similarities between mother's milk and infant stool. We assume those similarities occur because these mothers are breastfeeding. But when we look at the patterns of viruses that are present, most of the viruses, again, are bacteriophages, but we see that there's a relatively small number of myoviruses that are present in stool. Breast milk has a lot of myoviruses. If we look at the infant stool, it's similar to adult stool in that there's a lot of these cyphoviruses and few myoviruses. But the one thing that you might notice is that this is not an insignificant number of myoviruses. This is also not an insignificant number of cyphoviruses, so that there is some overlap in terms of the type of viruses that are present. And when we just look at when we look at breast milk, about 60% we find only in the mother. In the infants, about 65% we find only in the infant. But what we find in both is about 30%. So it looks like there's about 30% of these viruses that are shared. That's not the conclusion you'd probably draw from the previous figure, but pretty much all of the myoviruses and all of the cyphoviruses that are present tend to be the exact same ones in both breast milk and infant stool. Again, evidence that viruses can be easily shared even between genetically related individuals. There are some sort of weak points in what we look at in the viromes. In other words, when you look at the literature on bacteria in the microbiome, it's fairly obvious. For example, one of the first studies that you might recall is just looking at, hey, are bacteria in the mouth different from bacteria in the gut? It's such a simple question. It's one of the first questions that was answered early on in microbiome analysis. And it's obvious, right, that the bacteria in your mouth are different from the bacteria in your gut. We don't have those sorts of studies for the human virome. We finally decided to just perform the study, and we can show this really nice homogeneous group of viruses in the mouths of people, whereas this large sort of heterogeneous group of viruses in the gut. But clearly there are a demarcation between gut viruses and salivary viruses. So in a lot of ways, what we find in the virome does tend to mirror what we see in bacterial communities. One of the things that we're also really interested in is the idea of viruses and what we call classically sterile sites in the body. So we were the first group to show that there's a virome present in the urine. We showed that there's a virome present in breast milk. We did saliva, dental plaque, feces, all these different body types. But for myself as a clinician, there's sort of a holy grail, right? And that's your central nervous system. We're sort of taught at a very, the first day you go to medical school, right? Your central nervous system has this nice blood-brain barrier. Nothing gets across it, right? That's why most of us don't walk around with meningitis all the time, right? But, one of my thoughts is that, well, these viruses are everywhere else. I mean, folks have even done viromes of the air now, so the viruses are everywhere. So that probably even the central nervous system, by looking at cerebrospinal fluid, probably has a virome. So we actually set out to look to find whether there's a virome of the central nervous system. I probably wouldn't be showing you this data if we hadn't found a virome. But if you compare it, for example, with saliva, which is generally about 10 to the 7 to 10 to the 8th virus particles per milliliter, we did these sorts of studies for cerebrospinal fluid, and we found about 10 to the 4th. So it's much, much less. And sadly, we did try to do that experiment that I had told you guys about, but let's just take some CSF, stain it, and take it down to the electron micrograph to see if we can find viruses. But these viruses, there's so few of them that no, we weren't able to replicate that experiment that we did years ago. Interestingly enough, though, we recently took a look at the viruses in these subjects just to get a sense of, do we find viruses that are present in every subject? And the vast majority of these viruses that we did find in cerebrospinal fluid are individual specific, but we can find a few viruses that were present in every single subjects' cerebrospinal fluid. And we found somewhere around 3% total that were present in 10 or more of these subjects, which is half of the subjects that we looked at. We looked at the structure of some of these viruses. Honestly, looking at phages, fairly nondescript. You're just kind of looking at a phage structure. So, but these are the phages that are present in every subject. You know, we will be able to learn something from this. We just have to study it in a bit more depth to figure out what this is telling us. The one clear thing that we can find so far is that most of these viruses that are present in multiple subjects are these sort of lysogenic viruses, or viruses that live in the genomes of the bacteria primarily. And one of the themes really throughout a lot of the viral research that we do is that viruses that we think of as lysogenic, we think that they're, for the most part, just sitting dormant in these bacterial genomes. We tend to find these subjects, these viruses over and over at every single time point, suggesting that they're much more active than we have thought they were traditionally in the past. One of the other things that we can do now that we've looked at so many different body sites in terms of their viromes is that we can compare them. Because unfortunately with viral research, there's, you know, one group does one technique, another group does a different technique. We all do different techniques, unfortunately, which makes it hard to compare. But now that we've looked at these 7 or 8 or so body sites within the same lab, using the same sort of techniques, we can compare to see if there are differences, observable differences that we can see. So for example, one of the things we see about feces is this preponderance of microviruses, which are these single-stranded DNA phages. In saliva, we see preponderance of these cyphoviruses. We believe that that's why viruses are so persistent in saliva, because these tend to be lysogenic viruses. In the urine, for example, we found all of these herpes viruses and body fluids, which we haven't really talked about. We looked at, for example, bowel. We looked at joint fluids, et cetera. We find a good number of these inoviruses. In breast milk, we find all these myoviruses. The interesting thing is that looking at cerebrospinal fluid is actually fairly nondescript. If I had guessed before we did these experiments, I would have guessed that this would be the most distinctive of all the sample types that we looked at. It turned out to be amongst the least distinctive of all the sample types that we looked at. But trends are clearly occurring that we're starting to notice amongst these different body fluids, right? So for example, these two are basically showing the same thing on different axes, but if you concentrate on panel A, again, you see urine, which is fairly close to adult stool, which is fairly close, at least on the x-axis, to saliva. But then on the other side of this principal coordinates plot, you find all those other body sites: plasma, body fluids, CSF, milk, which seem to be somewhat similar. I'd say the one exception is that the milk seems to have characteristics that none of these other sample types have. So, you know, when we first looked at this, we'd say, well, let's just divide this in half. This is one cluster. This is sort of a second cluster of viromes that we're seeing. We next looked at the diversity, which basically is kind of telling you how many viruses are present and what is their distribution. And we also see a really sort of clear trend, and that is if you look at stool, urine, saliva, these are sort of high diversity sample types. If we look at CSF and plasma and milk, et cetera, these are relatively low diversity sample types. What do these have in common? These tend to have a lot of bacteria. These tend to have few bacteria, with the exception of the breast milk, which does have, which is generally somewhere in between in terms of the number of bacteria, but it's clearly showing up in this sort of low diversity cluster. So we've got these two different clusters, one with high diversity, one with lower diversity, that we could clearly see distinctions upon on principal coordinates analysis as well. Then we sort of looked at, well, which of these viruses are really shared, or which sample types can we find the same types of viruses over and over again. And if we look at body fluids, what we see is that it's very similar to what we find in cerebrospinal fluid, what we find in milk, what we find in plasma. If we look at CSF, It's very similar to what we find in body fluids, what we find in milk, and what we find in plasma. The same sort of trend for milk, which is similar to CSF and plasma and body fluids, the same for plasma. So there's a real big distinction here. If we were to look at the feces, we see that most of the fecal viruses look like they're shared with urine and with saliva. Most of the urine viruses are shared with saliva and shared with feces. In fact, even most of the salivary viruses, again, are shared with urine and shared with feces. Saliva does seem to be the slight exception, but it looks like you could almost just bifurcate this into, again, the low diversity cluster and the high diversity cluster of viruses based on basically what sample type you're looking at. And one of the big questions is, do we see this difference because we're just sharing one virus? You know, are we sharing that same virus in the mouth and in the gut and in the urine to give you that trend? And this clearly tells us that we're not. We're sharing viruses all across all of these different virus families and CSF compared to the other sample types within that cluster, and with stool compared to the other sample types within the high diversity cluster as well. The only exception tends to be that we share quite a few more viruses than we may have predicted between CSF and saliva, which we won't go into too much detail about. So what we think is going on is that there's these two different ecological clusters within your body, which you know, the diversity is shared within a cluster. Again, that's a high diversity cluster, a low diversity cluster. In general, body sites that have a lot of bacteria tend to have a lot of viruses. Body sites that tend to have few bacteria tend to have few viruses. And we believe, particularly when we look at these low diversity cluster, that most of these viruses get access to these compartments through transcytosis, which is a phenomenon that one of our collaborators, Jeremy Barr, who used to be right here at San Diego State, recently demonstrated. So now moving into why we're really become interested in bacteriophages over time. And that has to do with a couple of studies that we have performed that tell us a bit more about the ecology of these of bacteriophages in health and disease. So one of the earlier studies that we did was we were really just been wondering the question, hey, you know, you can look at periodontal disease, you can see that the bacterial communities in a periodontally healthy person are much different than the bacterial communities in a periodontally diseased patient. Is the same true for viruses? So we did this cross-sectional study, 9 folks with periodontal health, 7 with moderate to severe periodontal disease, and this pattern emerged really immediately, where we looked at supragingival, subgingival, and plaque and saliva, with subgingival plaque probably being sort of the sweet spot that you want to look at when you're interested in periodontal disease. But all the black are these patients in disease. All the white generally represent those patients with periodontal health. So there's a clear pattern that has emerged, and we can show that in fact this is highly statistically significant. Even in your supragingival plaque, which is the aerobic, the plaque in your mouth that's in the aerobic environment, the subgingival plaque is the plaque that's in the anaerobic environment, but they are distinctly different from the viruses you will find in saliva. The subgingival plaque viruses are distinctly different from the viruses you will find in subgingival plaque as well. And the big difference that we found, if we just look at the subgingiva, is again, we see this trend where there's a whole lot of cyphoviruses in the blue in healthy people. But once we hit sort of periodontal disease, all of these cyphoviruses appear to be replaced by myoviruses. In general, even though there are a lot of exceptions to this rule, cyphoviruses that we tend to find in the oral cavity tend to be lysogenic, and many of the myoviruses that we find in the oral cavity tend to be lytic, so that we're starting to wonder, well, what is the cause of periodontal disease? Are the viruses driving the disease, all of these myoviruses that we now have found? And it sort of really becomes a chicken and the egg. Are these changes in the phage community just a reflection of changes in the bacterial community, or are these phages actually driving the change? And that's a question we really haven't been able to answer in our studies. Another group has gone on to look, for example, at Crohn's disease and ulcerative colitis, and can even find virome differences in individuals who have ulcerative colitis. So it's not just periodontal disease where you can find those differences. Another study that we recently have started looking at again is a study we did a few years back where we were really interested in how diet affects your virome. And it's the only study we've actually done in mice rather than humans, but collaborating with Marty Blazer at Rutgers. He had this set of mice where basically he had just been placed them on a normal diet, but then at about week 16 had transitioned them to high-fat diets. And our big question is, does that transition to a high-fat diet affect virome contents? And the one thing that I can say, just like in humans, your gut is full of viruses. In mice, we see the exact same trend where they're just really highly enriched for all of these viruses that we can identify. Again, most of them are bacteriophages. One of the things that we know from looking at mice on normal and high-fat diets over time is that generally once you transition them to a high-fat diet, you significantly decrease the diversity of the bacteria in their gut. Interestingly, even though it's not statistically significant, we see a trend towards increased diversity in the virome in the gut when we swap them to high-fat diets. And when we look at the beta diversity through principal coordinates analysis of these over time, it's harder to identify the distinction when we're just looking at bacteria through 16S, but these mice, you can really tell the time apart and the diet apart quite easily. So these are the mice at week 4. They transition just a week later on the same diet. You can identify that group. And then once we hit week 5, you can identify further differences or cluster. But after we change out that diet by week 16, we see a much different virome in these mice. And by week 28 on that high-fat diet, they've transitioned even further. So what does this transition look like? Well, in general, you have a relatively stable virome when they're on these sort of normal diets. And just pay attention to, these panels show different things that we're not going to go into in detail. But most of these have relatively stable viromes over the course of week 4 or 5 to week 15. But the second we switch them to those high-fat diet, you can start to see changes emerge in the virome. And by week 28, these viromes are completely different than they started out. The big difference really are these microviruses. So again, these are single-stranded phages which seem to bloom when we put these mice on high-fat diets. And if we look at this same sort of information a different way, we can see that this bloom is highly statistically significant once we transition those mice to high-fat diets. But the other thing that we see is similar to what we saw in patients with periodontal disease, which is the cyphoviruses tend to be replaced by a different type of viruses. In this case, it's not the myoviruses. Instead, it's these microviruses. But we have basically the same question, right? Are the viruses helping to drive the differences that we see in the bacterial communities, or are the viruses just a mere reflection of the changes that we see in the bacterial community? And we haven't had good ways to be able to figure this out. We finally are performing experiments to try to determine that, but we've been working for the past few years on a different system to try to help us figure out and better understand what these viruses are doing to these bacterial communities and whether or not they're involved in driving these disease processes. And working with Dr. Emma Vercoe at the University of Guelph, she's got this wonderful system called chemostats, which I think many of you probably have heard about. But we used to use the term uncultivable or unculturable when it came to bacteria all the time. And what we figured out over the years is that there's probably very few, if any, uncultivable bacteria out there. We just have to find the right conditions to grow them. And her system really has become sort of one of those systems where we can grow all those bacteria where we previously thought we couldn't grow. And so the system they call the RoboGut, which is basically just a fancy sort of bioreactor where you control the temperature, you can control the nutrients and so forth, but they let it go for long periods of time so that when you put feces, for example, into the system, by the time you've hit day 24, the bacterial community that comes out actually looks remarkably like the bacterial community that you actually see in human feces, and it's full of a bunch of organisms that we used to think were uncultivable, but actually do grow quite well over time if you place them in the right conditions in these. So one of the things that we've been thinking about the system is it's reproducing the bacteria so well that's probably reproducing the viruses as well. So we actually got some of this material from them that they call liquid gold, and just ask the question, are there viruses present within this system? And when we look at feces, sometimes you'll find feces that's about 10 to the 8th. In this particular study, we found the feces about 10 to the 9th virus-like particles per milliliter. When we looked in the chemostats, they were the exact same order of magnitude. So these systems, which people have been using now for many years, are just teeming with viruses. Nobody actually ever looked to know that they were really full of viruses. And when we look at the viruses in these systems over time, they tend to mature once they hit day 24, which is about the stable state for these systems. So you get one sort of phenotype all the way through day 16, but when you hit day 24, the patterns of viruses that you see in all of these subjects that we looked at look remarkably similar to what you find in human feces. Even if we do sort of a basic principal coordinates analysis on this, we see that the liquid gold always groups together with the feces. So we're not only reproducing the viral communities, but we're reproducing this sort of individual specific nature that you tend to find in the human microbiome of different individuals. So we love this system. And one of the main reasons that we love it is that we want to take, for example, we can take your stool, we can grow something that looks almost exactly like your gut microbiome within this system, but not only are we reproducing the bacteria, we're reproducing the viruses that come from your gut as well. And what we want to do is, for example, take viruses from somebody else and put them into basically the gut microbiome of a different person and sort of ask the question, well, what happens, right? What happens on a daily basis when you shake hands with people or when you live with somebody and you're sharing viruses? So we think we have sort of the perfect system to finally be able to test that. And the benefit of that is that not only can we measure what's going on with these viruses, but these viruses have to compete with your own viruses within the system. So we're going to be able to reproduce the sort of competition that we know takes place in your bodies really on a daily basis. And that's sort of what we've planned to do, but we're early on and don't really have any results from any of those experiments. So in summary of all the things that we sort of have mentioned today, we know that human body surfaces have these really robust communities of viruses, most of which we identify as bacteriophages. We now know that these bacteriophages are really highly persistent members of your microbiome, and not only do they have body site specific, but they also have individual specific features that we can measure. We know that now that most, if not all, body surfaces have viral communities. We've looked at body fluid, CSF, breast milk, others have looked at skin. We now don't think that there's any site in your body that's devoid of these viruses. We also know that there are these high diversity and low diversity ecological clusters, which to some extent may be governed by the number of bacteria that we see, because most of the viruses we see are actually phages of the bacteria that are present. These phages are shared and they are transmissible between close contacts, unrelated close contacts. We also know that they're shared between genetically related close contacts as well. We can find these sort of ecological differences in the viromes in the subgingival crevice of people who have periodontal health and disease. We can also find, particularly in mice, that these viromes will respond to diet. And we think we're now poised to be able to answer the question, are viruses driving these conditions, or are these conditions basically driving changes that we see in the virome? And lastly, we've now got a model system with these chemostats that we can use to sort of assess the ability of viruses to perturb these communities so that we can understand finally what the role of these viruses may be in the human microbiome. So I just like to end by acknowledging current and former members of our laboratory, specifically Tasha Santiago Rodriguez, who's done, quite frankly, probably most of the studies that you guys have seen today. Shira Bellis did a bunch of these studies, as well as Refugio Robles, as well as our collaborators at Guelph, USC, IPATH with Chip Schooley and Stephanie Strachty, as well as our collaborators at Rutgers and our funding sources. So hopefully I'm not way over, because I'm happy to take any questions. Hi.
You mentioned that there are phages in the CSF, but how can that be the case if we know that the CSF can't ever have bacteria?
So the question was, how can there be phages in the CSF if we know that the CSF does not have bacteria? So a couple of different answers to that. I would say that I don't know that we're 100% certain the CSF doesn't have bacteria. I know a lot of researchers, including our own, who have taken CSF and done some standard things like 16S PCR, et cetera, and easily been able to produce products. But we're all too afraid to publish that information just because, you know, it would be so remarkable, the idea that there would be a bacteria in the CSF. The other thing that I would add is I mentioned the term transcytosis, which is the idea of sort of these bacteriophages sort of actively being taken up and moving across barriers. And you know, Jeremy Barr's group, I think, has shown fairly well that transcytosis occurs, so that we actually assumed that there would be bacteriophages in the CSF, mostly because we assumed that things like transcytosis were occurring, meaning that the viruses themselves don't necessarily have to have hosts in the CSF. In other words, when they make it to the CSF, it may very well be a dead end for them, but they make it in nonetheless because they're capable of escaping across through the blood-brain barrier. The one point that I would make that was really interesting to us is because we thought transcytosis was occurring, the largest surface area in the body is your gut, so that we actually assumed before doing these experiments that your CSF virome was going to look at least in part like your gut virome. It does not. And we were surprised to see that it looked a lot like your oral virome instead, if you guys notice some of those figures. You know, the oral cavity is a huge surface as well, but it still doesn't rival the sort of surface area that you have in the gut. Maybe it's proximity that allows them to get there easier from the oral cavity. We're unsure of what the answer to that is. Hi?
In the case of the stable viruses in saliva, do you know like which ones they are, and what do you think it's like their niche? Like, are the bacteria very stable, or are they like seeding from somewhere else?
So the question is, in terms of the stable viruses in saliva, really why do we believe they're stable? Are their hosts stable, et cetera? Yes, you know, the one thing that we know about bacteria and viruses in the human microbiome is that they compete. And there's a lot of different mechanisms by which they compete. Sometimes the bacteria is just going to, you know, develop mutations that are going to prevent the phage from infecting it. Sometimes it's going to use something like a CRISPR system. Sometimes they're going to use restriction modification system. The sort of competition that you see ongoing between bacteria and their viruses in the human microbiome is constant and daily. What to me is probably the most interesting part of that is that before starting to study phages and so forth, I just would have always assumed that there would be a winner. It looks like there basically isn't a winner, that many of these cases just result in a stalemate where these guys just continue to compete back and forth against one another over time, and that's why we continue to find these same viruses and these same phages. The other point that I want to make, because that's more of an argument I think, for lytic phages, is the high proportion of lysogenic phages that we see. In other words, phages that live in these bacterial genomes. I just would have assumed that phages would live in the bacterial genomes. They're not lysing their hosts very often, and that's sort of how they stay around. But because we find them at every time point in what we believe are relatively high abundances, I think we sort of had to reshape our thinking about that there probably is some low-level, I don't want to use the word constitutive, but lysis of these hosts that's occurring on a basically somewhat regular basis, and that's why we see these same things over and over again, because they're living in the genomes and lysing at some low level probably. Hi?
So I'm curious about the oral, the healthy and the non-healthy. Kind of two questions. One is, are you going to study those people longitudinally to see whether once the periodontal disease, I assume, resolves, whether they look more like the normal? And the second question really is, saliva goes through your gut, gets to your poop. So do you see connections in terms of, especially if you do that longitudinal study, you're going to see changes all the way through the gut too.
Okay.
I guess I'm just kind of curious, why aren't they looking similar
Okay.
than they are in saliva?
I have a hard time with remembering two questions at once.
Okay.
Let's go back to number one, and then we'll get to number two. What was one again?
One is, are you doing longitudinally? Do the people reverse?
Yeah. The question is, when you look at periodontally healthy versus folks with periodontal disease, when you look at them over time, as the periodontal disease improves, does their virome start to look periodontally healthy? The answer to that is we don't know. And it's something we would like to study. I have to say that dental plaque is not the easiest substance to get viromes from. It took us really quite a to do that, and we did something that we really hate doing, which is, because each tooth in the mouth is very individual and can have a much different microbiome on, different sides of the tooth, et cetera. So that to do that study, we actually had to pull plaque from different teeth in the mouth, which for us is a bit of a no-no, but we had to do it anyway to be able to do it. So the answer is that's a technically challenging set of experiments. We would love to revisit it at some point, but we haven't. Your second question is why.
What's the connection between the saliva and the poop?
Yeah. And the way I interpret it, correct me if I'm wrong, is, well, you're swallowing your saliva all the time. Why aren't you seeing, all the phages from saliva basically in your poop? And I think a lot of that has to do with, the body is just an incredibly competitive environment for bacteria and their phages. So if you think about it from almost a bacteria-centric standpoint, you could ask the question, well, if you're swallowing saliva that has all these bacteria in it, why aren't you finding all those same bacteria in the gut? And one of the main reasons is that these bacteria become incredibly well adapted to all the different niches in your body. So for example, as I was mentioning, your tongue is a different niche than the top of your tooth, which is a different niche from that which is below the gum line, which is different from your upper palate. They're all different. They all have different microbiota. So these bacteria have developed a very competitive landscape. And that competitive landscape is much different when you look at the gut. So for example, the most abundant bacteria you find in the mouth will be a streptococcus. The most abundant bacteria you'll find in the gut will be a Bacteroides. And part of that is just that the bacteria in the mouth aren't as well suited to be able to compete with all those organisms that inhabit all the different niches in the gut. And to a large extent, the phages that end up infecting and multiplying so that we can find them in the virome are a reflection of the microbes that we find in the different locations. Okay?
What do you make of the study that suggested that this one particular bacterium, P. gingivalis, is associated with Alzheimer's?
Yeah.
And are there phage for that? And is
Yeah.
that whole
Yeah.
line of
So the question is, what do we think about P. gingivalis in a single study being associated with Alzheimer's? And I will have to say I haven't seen that study yet, but it does not surprise me that a study like that can come out because there's all of these sorts of association, periodontal disease and coronary artery disease and periodontal disease and Alzheimer's and so forth. So it doesn't surprise me that someone's been able to find an association. I look at it more from a perspective of, is there some sort of causation? And without having evaluating the study, it's really hard for me to say. Are there P. gingivalis phages? I believe the answer to that is yes. I don't know that there's any good ones yet. I know I've had several conversations. I've sat on dental study sections about P. gingivalis phages, but right now the answers to that are kind of escaping me. But I do believe there are available P. gingivalis phages. I just don't know how good they are, et cetera. Okay. Oh, one other question. Hey.
Very interesting topic, David. So you showed a lot, especially difference between healthy disease or even healthy cases, and you identified a lot of the players, and the players were changing. But a lot of the nuances, the abundance of the viruses, and are they changing? Where are they coming from? Are they coming from within, or are they coming from other sites of the body when a new disease happens? Or how do you explain cases like when you do the mouse study where everything's kind of sterile, but now we get all these kind of new viruses coming from somewhere?
Yeah. I think the question is, well, we showed a lot of different things. How do we explain changes in abundances of viruses, particularly looking at the murine study that we showed where you give a mouse a high-fat diet and suddenly there's a bunch of microviridae. I will say that in particular on that point, I'm not sure if this addresses your entire question, but those microviridae were always present, So in other words, those microviruses were present in the individuals that were on normal diets. They were a tiny proportion of the community, so you could barely even notice them on those charts, and they just bloomed once the diet changes. So that we don't believe that these are new viruses coming into the community. They're just viruses that are already there that are blooming. And the question is, are they blooming because their hosts are blooming? We don't know what their hosts are, which is upsetting not to be able to know that. That's one of the reasons we're kind of moving into sort of more basic phage things so we can identify a lot of these hosts. But are they blooming because their hosts are blooming, or are they blooming because other factors might be causing them to bloom? So that's really for us the really the big question, because we don't, in a lot of our studies, if we sequence deeply enough, we don't really believe that we're identifying a whole lot of new stuff. I think the core virome in general in most of the studies we've done is about 70% of what we find just ends up being the same stuff, whether relative abundances change or not. Then there seems to be this sort of 30% that tends to be more variable and is more susceptible to change. So that 70% we don't see change that much. It's the other 30% where we see most of the change. That's in humans. But we didn't see much of a difference between mice and humans. I'd say the one big difference we saw between mice and humans is that these mouse viromes were so much more diverse than we've seen in humans, which, I mean, if you think about the surface area of a mouse gut compared to a human gut, that for me is a really sort of big surprise. All right. Very good. Oh, one more.
In spinal fluid, I'm fascinated about the viruses that you find. And I'm assuming that you spin down the CSF and then sequence what's left of the CSF?
So we did these by a technique that we don't do anymore, but it's the same technique that Forrest Rower tends to do here. which is first we filter things fairly heavily to get rid of cells that we can. Then we put them on cesium chloride gradients. And we take the fraction where most known viruses are. We take that fraction, we then we'll use a DNase to try to lyse any naked DNA. Then we'll, burst the virions and sequence whatever DNA is left over.
Because there's always a few cells in the CSF.
Yes.
And if some of them die in the CSF, and there's genomes, host genomes that may contain stuff like herpes simplex virus as latent.
Yeah.
I'm sure there's a ton of other viruses that infect our cells that
Mm-hmm.
we haven't yet discovered. But would you pick those up on this?
Yeah.
assay?
So let me repeat your question just for if anybody's streaming, because I forgot to do that. I think the question really is, how are we isolating viruses, and is there a chance that we're seeing sort of either contaminating cellular material or viruses that are present in contaminating cellular material? In general, it's really hard to deal with the idea of viruses present in contaminating cellular material, right? Because when we filter it well. So we put it through multiple filters. We're not able to grow things, and we certainly don't find any human DNA in the viromes that we look at. But if there's a cell that's lysing, that's full of herpes, and that herpes comes out before we filter it, that's going to look like it's part of the virome. And we don't have any great way of controlling for that. And the same thing, for example, with bacteria, right? If we're filtering out bacteria, we don't think there's bacteria in the vast majority of these CSF specimens. If they end up lysing and releasing their viruses before we filter them out, then we're looking at viruses that actually weren't free to begin with. They were actually cellular. So that's a potential problem with any sort of virome analysis. I think there are some groups that, in fact, Forrest Rower's group has shown that you can take, for example, a sample type, you can put an induction agent on it to make the viruses come out. And I can tell you those viromes that have been induced look a lot different than the viruses that we look at. That doesn't mean that there's zero induction in our viromes. It just means that we're probably not seeing, at least not that large scale induction when we look at a virome. Oh goodness, more questions. Okay.
So you showed like a big uptake in microviridae under like disease conditions, so like oral disease. Does the fact that, so presumably a lot of those viruses get washed into the gut, the fact that then you don't have disease developing in the gut with this like large amount of microviruses being washed down is safe?
Yeah.
I guess one is like that there's different hosts in the gut, so they're not having an influence.
Yeah, I'm not certain I fully understand the question. I'll sort of repeat what I understand about it, and that is that we show a lot of microviruses in disease, and do those become washed away, or when they make it to the gut, do they find hosts? Is that what you're asking?
So in oral disease, then you find a lot of microviruses.
Now, so in oral disease, we don't tend to find a lot of microviruses. If you remember from some of the slides, microviruses that we found tend to be much more abundant in the gut than any other body surfaces. So what we found in oral disease was a transition to myoviruses, not microviruses. But if the question is could there potentially be hosts for those myoviruses in the gut? Sure. It's a problem when you don't know what the actual hosts are, because when you're asked could there be hosts in the gut, the answer I think has to be sure until we know more information about them. Again, your question I think highlights why a laboratory like ours is starting to try to grow viruses, because the question that's been out there since the start of all this virome work is what are the hosts for all of these viruses? And having to say we don't know over and over for years has really pushed us to try to figure out exactly what they are. All right. Hey? Oh. Thank you very much.